Good afternoon, everyone. Well, it's great to be here with you virtually today. My name's Armin Raznahan, and I'm a child and adolescent psychiatrist. I lead the clinical research team at the NIMH just outside Washington, D.C. And XY variations are a major focus of our work. So what I'd like to do today is tell you a little bit about our research, what we're trying to achieve, what it's like to participate in the study, share a couple of our recent findings, and tell you about our future plans. Before I dive in, though, I'd like to give two big shout outs. The first is to you all, AXYS and the wider XY variation community. We're really grateful for the support you've shown our research over the years, and none of what I'm about to show you will be possible without your support. I hope you approve of the research directions we're taking. We'd really like to hear from you if you think there are more important directions that we're overlooking. The second shout out is to the wonderfully vibrant community of clinicians and scientists that are such fabulous colleagues to work with in this field. It's been really exciting to see more and more people paying attention to XY variations as the meeting grows over the years. Okay, so the 5 things I want to do today is give you a quick overview of the current NIMH study on XY variations, how it came about, and particularly how we're responding to COVID. Who's on the study team and what are our main goals? What's the study design? What is it like to take part in the study? And then I'm going to dive into a couple of recent findings from our research and then tell you who to contact for more information. So let's start at the start. So the NIH XY variation study has been running out of the NIH Clinical Center that I'm showing in this picture here for over 30 years now. And it's a 2-phase study. So the first phase ran between 1990 and 2010, and it was initiated and headed by Dr. Jay Geed. And this study it recruited about 150 participants with varying XY variations and focused particularly on cognitive development and brain anatomy. The second phase of the study is more recent and has been running out of my lab for about 6 years now, still at the NIH Clinical Center. And what we wanted to do in this phase is significantly expand beyond the first phase of the study in 4 major directions. So first, this current phase is over 3 times the size of the first phase, and we're funded longitudinally too. So we have resources to see about 500 participants at every time point and follow these participants over time. A second difference is that we're gathering many more detailed measures of cognition and behavior from the participants in the study. And this is for a couple of goals. The first is so we can provide richer clinical feedback to the families in the study. And the second is to help us better address some of the scientific questions that we're trying to go after. The third difference is we're not just gathering information on the participant themselves who has the XY variation, but also on their first-degree relatives, because we think this information may help us predict outcomes a bit better. And I'm going to give you some examples of that later today. And finally, above and beyond measuring cognition and behavior, we're really investing much more heavily in getting more detailed measures of the brain and of the genome. So for example, we're not just studying brain structure, but also brain function and brain connectivity. And there's also a major new strand where we're trying to characterize how XY variations impact cells and the functioning of the genome. So one thing that's very much shared between the current phase of the study and the first phase is we don't just focus on one XY variation. We gather this same detailed information I've just described on all XY variation groups. So far, we've completed the first time point for seeing families with XYY syndrome and XXY syndrome. And we just started seeing trisomy X families when the pandemic hit. And I just want to take a few moments to talk about the pandemic. It's obviously impacted all of our lives in many ways, as represented by the fact that we're having this meeting virtually. So I want to tell you a bit about how it's impacted the research and what we've been doing to try and keep the research moving forward in sort of COVID-proof ways. So our study paused in March 2020, the on-site version of the study, because patient enrollment into the NIH Clinical Center was paused. And we just started seeing trisomy X families at the time, as I was saying. The good news is we hope that NIH is opening up again. We're anticipating that sometime this fall or winter, there'll be a gradual return to on-site studies for study participants, and we're ready to restart the on-site study at full speed the minute that happens. And we'll begin again with trisomy X families and then work through all the other XY variations subsequently. So we're poised to go as soon as we get the green light, and we hope the green light for on-site studies will come later this calendar year. In the meantime, though, we've set up 3 major initiatives that are designed to bypass COVID, to enable us to continue the work without requiring participants to physically come to the NIH while that's prohibited by COVID. So we've been developing, and we're about to deploy in the coming weeks, a new virtual version of our study, which will enable families to interact with the clinical study team remotely without leaving their homes. We've also made major new investments into cellular research. So we already have collections of blood and skin tissue from participants who come through the study, and we've been using these to make brain cells in a dish that we can study without requiring the patients to physically come to the NIH. And I'm also really excited to announce that we've teamed up with other NIH colleagues to start a new hunt for genes that might influence why some people seem to be very severely impacted by an XY variation, while others seem to experience very few effects at all. You would have already heard from Mike Seltzer, a member of this team, the clinical genomics team from NIAID, and I'll give you some contact details for our collaborators in this team later on in the talk. And we've continued throughout the pandemic to do research with the data that you so generously shared with us. There have been about 10 papers that have come out in the last year that relate to XY variations, and we can provide access to all of those to AXYS and to the wider community. I'm giving you a couple of examples here, and I'll be digging into some of them in detail later on in the talk. So who's on the study team and what are our main goals? Well, here are some current and recent members of the study team, and many of you will be familiar with Jonathan Blumenthal, our project coordinator, and Erin Torres, our fabulous psychiatric nurse practitioner. Our clinical team also has several highly specialized developmental psychologists from Children's Medical Center, myself as a child psychiatrist, and a large numbers of trainees representing diverse disciplines. And this clinical research team works really closely with a behind-the-scenes data science team that uses computational approaches to try and understand the masses of clinical brain and genomic data that we gather through the protocol to try and advance our understanding of XY variations. And we also work very closely with a large team of collaborators in America and in North America and Europe. So we all work together towards this goal of using data to advance clinical care. And the research we do can be thought of as being organized around key questions or concerns that we often hear from families who come through the study. So for example, we commonly hear families say, we had to tell the doctor what trisomy X was, for example, this experience of meeting care providers for the first time who might not be that well informed about XY variations. So what we've been trying to do is to really increase awareness of XY variations amongst clinicians and scientists, particularly within our local community of mental health researchers, so it's higher on their radar. We're also publishing papers that try to describe in fine detail what some of the difficulties that families affected by XY variations can have, and to share this information widely with clinicians. So hopefully, pardon me, the clinicians who become aware of this work are a bit more informed when they meet their next family. Families can also often ask, what can we do to support our child's development? Should we consider treatment A or treatment B? And I think the answer to these questions really comes back to fully describing the very wide range of outcomes that one can see in individuals and families affected by XY variations. So a lot of the work we do tries to describe just how diverse these outcomes can be and figure out what the factors are that might predict how severely impacted a particular individual might be. And then finally, families will often ask, why is our child having these developmental difficulties? In what way are extra X or Y chromosomes affecting their brain development? Are there tests we can take early on that predict what might happen down the line? And I think to answer these sorts of questions, we need to better understand how XY variations impact brain development and impact function and structure of the genome, and then to link these biological features up to the cognitive and behavioral aspects that impact people on a day-to-day basis. So what's our study design then? As we're trying to reach these goals, how's the study structured and what's it like to be a participant in the study? Well, before I dive into what the study looks like, I just want to identify 4 kind of high-level guiding ideas that organize the way we do things. The first thing is we believe that we'll do a better job of understanding any individual XY variation if we study all XY variation groups. So if you want to understand how carrying an extra X chromosome in XXY syndrome might be affecting development, for example, that question is really assisted by understanding what's happening in trisomy X and vice versa, for example. We also think it's really important to understand in individuals as well as groups. As I've mentioned before, there's huge variability in the outcome that we see in participants through our study. And that's really what we think we need to understand. Where is that variability coming from? I think we also believe we need to get much better at measuring development. So history has given us these standard questionnaires that we might ask it, we use in the clinic to characterize someone's experience, but we think we can improve upon these questionnaires, and I'll give you some examples of that a bit later. And then finally, I've mentioned this a little bit already, but I think it's really important to try and build a linked up understanding of how XY variations impact development. Very much so at the level of cognition, behavior, and learning, which impact day-to-day functioning, but then to try and figure out how these impacts relate to the way XY variations are influencing the brain, and how the brain impacts relate to the way XY variations are influencing the genome. So I'm giving you this sort of approach across the 3 levels here. So we've done work, for example, that characterizes sets of genes that alter their expression in individuals who carry extra X chromosomes. We've also, at the next level, described sets of brain regions that change in their size as individuals carry extra X and Y chromosomes. So these green regions change their size with individuals who carry an extra X. The blue regions change their size as a function of carrying an extra Y. And the red regions change their size as a function of carrying an extra X or an extra Y. One can also take these data science approaches to thinking about behavior. So each of these circles is a different scale we have to measure cognition and behavior in XYY syndrome. And although there might be 20 individual circles here, we can use computers to identify subsets of circles that tend to hang together. So rather than measuring all 20 circles, these results tell us we may only need to measure 3 areas: general cognition, mood and early milestones, and autism spectrum-like behaviors. So in order to do research that spans analysis of cognition, behavior, the brain, and the genome, what kind of research study do you need to make this happen? And that maps onto what's it like to be part of the study. Well, this is giving you a schedule for, in this case, it happens to be a visit for an XXY family. Families will come to the DC area for 3 days or more, and 2 of those days will be spent at the NIH Clinical Center. And they'll involve multiple different elements of the study. So there'll be a welcome and orientation, gathering basic physical information, detailed psychological testing and autism spectrum disorder testing, practicing to go into the scanner, cognitive testing with the family members, measures of physical development, a detailed interview for day-to-day functioning and mental health, and then a significant portion for MRI scanning, and as I mentioned, detailed cognitive testing in the participant and their family members. And we take all of this information and it serves 2 main purposes. The first is for each family that comes through the study, a multidisciplinary team of clinicians sits and discusses all the information we've gathered, pulls it together into a single report that provides a sort of summary of how the young person is doing and makes some recommendations for future care. And that report is given to the families, and it can sometimes help to access services locally where they live. The second major output is that all of these data are stored on secure servers and used in computational analyses that help us better understand how learning, cognition, behavior, the brain and the genome are impacted by XY variations. So this is the on-site version of the study. which will be starting again, hopefully, as I said, later this year. But in the meantime and in parallel, we're running a new virtual version of the study. So families will get a link, which they just hit by email, and it takes them to an online questionnaire that they can complete on their tablet or on their computer at home. It gathers basic demographic information and also detailed measures of strengths and difficulties that the young person might be experiencing. So in the remaining time, I just want to touch on a couple of recent findings from the study. Now, the examples I'm going to give relate to the XYY phase of the study, but I'm sharing them here because they very much indicate the way we think we need to be doing our work for all of the XY variations. And I think many of the general principles that you'll see here in relation to XYY syndrome are going to be relevant for the other sex chromosome aneuploidy groups too. So one question that we've been tackling recently is, can we do any better at predicting how a specific child with an XY variation will be impacted? So currently, if a child is diagnosed with an XY variation, say XYY syndrome, for example, and the parent asks the carer, well, what's the likely outcome? The carer will look at published reports and find what the average outcome is in that group, for example, an XYY variation. And for example, the average IQ in XYY variation might be 90 as compared to the population average of 100. And that number of 90 will be what is fed back to the family to give a kind of rough idea of how development might be altered by XYY variation. But we're all individuals, whether we have an XY variation or not. We all differ from each other. And understandably, what families really want to know is not what is the average impact of XYY, but how is this going to impact my specific child? So I think we need to get better at this prediction. And we were motivated by the idea that because people in the same family tend to be more similar to each other than people from different families, we asked, well, maybe we can use measurements in the family members of the affected individual to predict how likely the impact, how severe the impact is likely to be for the young person in question. And I'm showing you that in this graph here. Let me just walk you through it. So on the x-axis, we have the family score in a test of vocabulary. 10 is the population average. And on the y-axis here, we have the child score. So each of these dots represents a child and the average score for their first degree relatives who don't have an XY variation, mum, dad, and siblings. So here, for example, is a child who has a vocabulary score a little bit above the population average of 10, about 14, and their family have a vocabulary score of about 15 on average. Whereas here you have a child with XYY who has a vocabulary score of about 2.5, and their family has a vocabulary score of about 7.5. So if all of these dots were on this line here, that would mean that the children's score will be exactly the same as their family score. The fact that most of the dots are below the line means that in almost all cases of XYY syndrome, the vocabulary score seen in the child is less than the average vocabulary score of their first degree relatives. But there's huge variation here. You can see it ranges from sort of 15 down to 2. And there are children with XYY syndrome who have above average vocabulary scores, greater than the population average, and others that have scores below the population average. And what this slope, this purple line, tells you is that if we know the family score, it enables us to make a more precise guess of the score that their child is likely to have, so that you can do better at estimating the impact in a child if you know something about their family scores. And this is for vocabulary. So just to recap what I've said, in XYY, but we think this is going to apply in the other XY variations too, we found that some aspects of thinking and behavior are more closely related between children and their family members than others. Where these relationships exist, it means that we might be able to use measures in family members to make a better guess at where the child is headed. What's really important is this does not mean that there's no potential for change, but if we identify a child who might be predicted to have a more severe impact based on their family scores, it might help us prioritize them for extra input early on and hopefully improve the outcome later on in development. We need to do some more work though to figure out if we can really use this technique in real time in the general clinic, but that's something that we're working towards hard. So a second question we've asked ourselves are, what are the main dimensions of behavioral features in XY variations, and how do these relate to real world outcomes? Mental health is such a complex and difficult thing to measure. Can we find the main dimensions and focus on measuring those? So we took 70 different measures of behavior and cognition from traditional questionnaires in the field of mental health research, and we gathered scores on all of these 70 dimensions, in this case in individuals with XYY syndrome, but as I say, it can be applied to other groups too. We took these measures and we submitted them to computers. Computers crunched the numbers, and they told us that these 70 different measures can actually be described by a much smaller set of just 10 features. So that the complexity of mental health difficulties that you can see in XYY variation can be described as scores on a smaller number of 10 dimensions: how you get on with peers, mood difficulties, anxiety, for example, to what extent you have difficulties with attention. And the important thing is that these different dimensions relate differently to real world measures that families really care about. So for example, if you think about day-to-day functioning, how good a person is on self-care, using public transport, cooking meals for themselves, getting on at school independently, it seems that attentional functioning in the young person is quite closely related to day-to-day functioning. What's helpful at predicting how good your day-to-day functioning is how good the child's attention is. But it's not the same for caregiver strain. Externalizing behaviors, a tendency to get into arguments or to break rules, isn't so strongly predictive of day-to-day functioning, but it's really strongly predictive of caregiver strain. So just to recap what I've told you, we can simplify the complexity of mental health presentations in XYY syndrome and other aneuploidies, we think, and other XY variations, into a limited number of dimensions. In this case, there's just 10 dimensions. It seems that ADHD-like and autism-like issues appear to be two really important dimensions out of these 10. But the different dimensions are important for different outcomes. So day-to-day functioning is more closely related to attention, whereas caregiver strain is more closely related to externalizing difficulties, kind of conflict behaviors in the young person. We think this research is helpful for a couple of reasons. One, it helps us speed up clinical assessment. We don't need to measure all 70 dimensions. We can just measure a smaller number of dimensions. But we think it also helps us figure out which difficulties we need to target for treatment development. It's not necessarily those aspects of mental health that are most severely impacted in an XY variation that one might want to go after. You may want to say, well, what aspects of mental health difficulties really predict how they do with friends, how they do with school, how they do in the family, in the household? And those are the things that you want to target. And the sorts of analyses I've just shown you help us find those targets. Well, I hope that's given you a general overview. This slide tells you who to contact for more information. I'm showing Jonathan Blumenthal and Erin Torres from our group here, and Riley Duncan from the NIH Clinical Genomics program that I was mentioning, our collaborators who are working with on this hunt for genes that might influence the severity of outcomes in XY variations. Please feel free to contact me, Jonathan, Erin, or Riley about these aspects of the study. I know they've been presenting throughout the meeting and are definitely interested to hear from you, so do reach out. And then finally, most important slide in some ways is to thank all of you and to thank each and every individual within our own research teams, our collaborators at the NIH and beyond the NIH who've helped to make this work possible. And thank you very much for your attention.